So now we come to our final stage of cellular respiration. And this is the chance for us to take all of those piggy banks, all of those NADHs, and cash them in for some ATP. So let's recap where we've gone already, what's led us to this electron transport chain. So as we can see, we start off with glycolysis, where we started to oxidize our glucose. We split it in half down to pyruvate, which allowed us to produce a couple ATPs down here. And we also produced a couple piggy banks. Well, we said, hey, there's still a lot of energy left in pyruvate, so let's shuffle it over into the mitochondria and continue to break it down. And during the Krebs cycle, we broke down that pyruvate down into carbon dioxides, and we once again squeezed a little bit of ATP out of it. But we also produced 10 more piggy banks, 8 NADHs and 2 FADH2s. So now we stand here with 10 NADHs, 2 FADH2s, 12 piggy banks, and only 4 ATPs. But the electron transport chain now is going to let us do something called oxidative phosphorylation, where we oxidize our piggy banks. We reduce oxygen, which becomes water. And more importantly, that process allows us to create about 36 more ATPs, about 3 ATPs for each piggy bank that we started. So let's see how we can make this ATP. So we've got two ATPs from glycolysis, 2 from the Krebs cycle, 4 ATPs. There's nothing to get too excited about, especially if when working muscle is going to use over 10 million a second, we got to have more. So enter the electron transport chain. So as we mentioned before, we're inside the mitochondria. We're inside that inner membrane, right? With all those folds, those cristae, right? Increasing the surface area. We have all these proteins, these enzymes embedded in there that are going to help us to transport electrons, right? And in doing so, we're going to try to make some more energy. And so we can almost think about this as kind of being a downhill flow. Ultimately, what's really happening is oxygen's at the end of this chain, and oxygen being super electronegative is pulling really hard. It's pulling those electrons to it. In other words, oxygen wants to be reduced, and thus that is causing these piggy banks, NADH and FADH2, to be oxidized, to give up their electrons. And we're hoping that by doing this final method, we're going to be able to take 1 molecule of glucose, 1 molecule of C6H12O6, and produce somewhere between 36 and 40 ATPs, almost 20 times more efficient than glycolysis alone. So there's our mitochondria. Again, we're going to be in this double membrane area, or this inner membrane area over here. That Krebs cycle, recall, was in the matrix, right in the middle of the mitochondria, creating most of these piggy banks, which then can get put right into this electron transport chain in this inner membrane. In between the inner and the outer membrane, we're going to be talking about some hydrogen ions accumulating. All right, so here's a brief look at our electron transport chain. Notice these blue lines. This would be our outer membrane of the mitochondria along the top. These little folds here, including the electron transport chain, is our inner membrane. At the bottom, we're in the mitochondrial matrix. This is the intermembrane space, kind of in a lighter gray, okay? And notice we can see a bigger picture of that mitochondria down below. We're not gonna worry about this dehydrogenase and cytochrome and all these kind of things. We're not gonna name those transporters or Q and C. Instead, we are just gonna worry about the big picture idea. What came in, what came out, what are some important things that happened in between? So, what did happen in between? Well, we've got our electron carriers. I think we remember them, right? Because who could forget a piggy bank like this? And so glycolysis, we made 2 NADHs. Krebs cycle, we made 8 NADHs, 2 FADH2s. We got 12 piggy banks sitting there. Remember, we filled them with electrons. We filled them with hydrogens. We're kind of using those interchangeably. We reduced them, in other words, right? And now we want to oxidize them. We want to cash them in. So, where do we do that? Well, a couple things to remember. First of all, you have to remember that a hydrogen is an electron plus a hydrogen ion, which is a proton. An electron plus a proton. There are no neutrons. So if we take a hydrogen, we could split that hydrogen into a proton and an electron, right? And remember that this NADH is going to give up its hydrogen, right? It's going to get oxidized back into NAD+, which can keep doing this whole process. The H is going to get split into an electron and a proton. And watch what happens in the electron transport chain. The electron gets passed through the chain. Follow the green electron. It gets passed from one thing to the other. And who's one of the most electronegative, electron-wanting things? Oxygen, waiting at the end. It's pulling these electrons to it. Well, what happened to the other part of the hydrogen? The H plus, the proton, is actually getting pumped into the intermembrane space. And so all of these hydrogens are accumulating in the intermembrane space. It's creating a concentration gradient there, a high concentration of hydrogen ions. So once again, NADH right here drops off the hydrogen. The hydrogen splits into the electron, which travels through, and the proton, which gets pumped into the intermembrane space. Now notice that FADH2, I've referred to this as kind of like a smaller piggy bank. Notice it drops off its hydrogen a little bit later. Therefore, it doesn't pump quite as much hydrogen across, and therefore it's not worth quite as much ATP. One way to think of it is NADH goes through 1, 2, 3 proteins and produces 3 ATPs. NADH2, on the other hand, goes through 1, 2 proteins, so it forms more like 2 ATPs. Okay? Again, not super important, but it does provide a little bit less energy. So, electrons are passing through. Electrons deal with energy, and as these electrons are moving through, they are releasing energy. And think about their energy is being used to pump hydrogen into that intermembrane space. And remember, that would be a type of active transport because you're forcing it into a high concentration on one side and a low concentration on the other. So it's active transport. The energy is being fueled by those electrons passing through. And then you'll see in a minute, once we have this big concentration gradient, we can let it go back through ATP synthase, and that's where we're going to actually make ATP. Okay? So the electrons are powering these proton pumps. The electrons are providing the energy to pump hydrogens across that membrane. So we've seen our electron carriers, right? We're cleaving or chopping the hydrogen off of the NADH and FADH2. Just kind of think about pulling those H's right off the acronyms there. The electrons are going to be stripped from the hydrogens, leaving a proton, H+. The protons are going to get pumped across against their concentration gradient into the intramembrane, forming a big concentration of hydrogen up there. And again, it's these flowing electrons as they move through the electron transport chain. The flowing electrons are what's providing the energy to do that work, to do that pumping of things across. And so, okay, big deal. Like, why are we creating a concentration gradient on the inside there or in the intermembrane space? It's so that, looking at this right-hand picture, it's so that the ATP synthase that we saw earlier in this unit, ATP synthase can use that concentration gradient and actually use that desire of hydrogen ions wanting to diffuse back into the matrix to actually recharge our ADP into ATP. So, why are these electrons moving? Again, that comes back to this idea of oxygen, right? Oxygen is super electronegative, most electronegative biological element there is, and it's pulling these electrons. So NADH is releasing them, right? It's splitting the hydrogen, proton, and electron, and oxygen keeps pulling those pulling those electrons, pulling those electrons, pulling those electrons. And again, as those electrons flow, their energy is released, building up, pumping hydrogens into this intermembrane space. Now, if you have an oxygen, what happens to an oxygen when an oxygen gains a hydrogen? Well, what do we know that's made up of oxygens and hydrogens? Water. And so in the end, oxygen is getting reduced. It's grabbing those electrons as they pull in. It's also grabbing the hydrogen ions that are going to get pumped back in. If you notice this hydrogen ion coming through ATP synthase, now it's inside. The oxygen grabs that. It grabs an electron. That's a hydrogen combined. Hydrogens plus oxygens give us water. So here's where oxygen is used and water is produced. And we're going to see this whole idea is called of making ATP with a concentration gradient is called oxidative phosphorylation at the bottom of your screen there. Oxidative phosphorylation. Rather than what we did with substrate level in glycolysis and the Krebs cycle, where we transferred a phosphate from one thing onto an ADP to become ATP, this process uses a concentration gradient to create ATP. And so think about these electrons as they're moving through that electron transport chain. Think of them as falling downhill. All right, think about this electron bouncing down the steps. Or don't actually try this at home, but think about yourself falling down those steps, right? What would you do? Well, I'm guessing you would release some energy, right? You'd be screaming on your way down. You'd be making some noise as you bump and thump and break your head open falling down. You'd have less energy at the bottom, right? You'd maybe stop screaming by them than you had at the top. You'd be releasing energy along the way. That's what's happening to these electrons as they're getting pulled down the staircase towards oxygen. They are releasing their energy. And that energy then, again, is allowing those pumps to pump hydrogen out of the matrix and into the intermembrane space. So it's like they're flowing downhill. Oxygen is pulling them very, very hard, right? And that's controlled oxidation. It's happening slowly in steps, right? Even in the electron transport chain, we're talking about small steps here. And so we're able to capture that energy rather than causing just a bonfire to happen inside our cells. So this is what it all comes down to. The reason why glycolysis and Krebs cycle and even all of that electron transport chain happened was so we get here with a concentration gradient of hydrogens in the intermembrane space. And so now we have all these hydrogens in the intermembrane space, very few in the matrix. And what did we learn about diffusion last unit? Things want to diffuse from high to low, right? It was one of our laws of thermodynamics. They want to diffuse in. And so this molecule called ATP synthase sits there and it says, sure, you can come through me. And it will allow them to pass through the middle. And when they do, look at these arrows here, when they pass through, they actually cause this complex of proteins to actually rotate, to spin. They're actually turning this concentration gradient, which is potential energy, into kinetic energy. And that kinetic energy is providing the energy then to combine an ADP diphosphate with an inorganic phosphate. These are those phosphates that are just floating around in your cells. Becomes ATP, triphosphate. And that's the idea of oxidative phosphorylation, or even chemiosmosis, using this idea of a concentration gradient to build our ATP. And again, this thing's called ATP synthase because it synthesizes ATP, and it's an enzyme that's doing this. This is where those extra 32 to 36 ATPs are actually formed. And this process, while we're referring to it as oxidative phosphorylation, is called chemiosmosis. Chemiosmosis. It's this idea of building this concentration gradient of ions and then allowing those ions to flow across a membrane in order to build ATP. Okay? So chemiosmosis and oxidative phosphorylation, for our purposes, I think we can use those terms interchangeably. We're building up the concentration gradient here, allowing it to go through ATP synthase, and that's what's making ATP. So when you take this idea, the electron transport chain is specifically these NADHs and FADH2s releasing their hydrogens, allowing the electrons to pass through, which is pumping the hydrogens into that space. That would do no good if we didn't have a molecule like ATP synthase that allows those hydrogens to come back through, which allows us to channel that energy to make ATP, to recharge ADP into ATP. Peter Mitchell is the one that figured this stuff out, but I'm not going to say anything more about him right now. So here is a little bit more of a complicated look at it, but it tries to combine a few extra points here. So here's our Krebs cycle over here, cranking out some NAD and FADH2, along with our CO2 and a little bit of ATP. Notice the NAD and the FADs, they release their hydrogens. Those hydrogens are split. The electrons are what's following the red line. The hydrogen ion part, the proton, is getting pumped into the intermembrane space, as we see kind of in the lighter gray. As we build up a concentration gradient there, hydrogen ions are allowed to go back through ATP synthase over here on the right-hand side. Meanwhile, you have oxygen guiding this whole thing, pulling these electrons through. You can see the E coming right into oxygen there. And it also grabs onto these hydrogen ions, these protons that are coming in. And oxygen plus a proton plus an electron equals H2O. And so in the end, we're able to take our 12 piggy banks, we are able to oxidize them by reducing oxygen into water, and that allowed this concentration gradient to then make many, many, many more ATPs. And so in the end, if we tie it all together, your poster will look similar to this, except you'll have more details for each of the 3 steps. But we can see we made 2 ATPs in glycolysis, 2 piggy banks, 2 ATPs in Krebs cycle, 10 piggy banks, because we have this one over here, 10 piggy banks. All the piggy banks go in the electron transport chain, and by oxidative phosphorylation rather than substrate level, we create about another 36 ATPs, giving us a max amount of 40 ATPs from one molecule of glucose, one molecule of C6H12O6. So now that we've gotten through this, these are some questions that you should think about. So you probably should pause right here, see if you can answer those questions. We'll give you an official sheet and some questions on the cellular respiration overview worksheet. We'll officially have you answer these questions. Now, one last thing. Think about what happens if oxygen becomes unavailable. If oxygen becomes unavailable, it can't pull those electrons. And if the electrons can't get pulled down, you can't oxidize the NADs and the FADH2s, which means you can't empty them. And if you can't empty them, those piggy banks fill up and everything else starts to back up, right? And everything starts to run out and you can't make ATP. And if that all happens, well, you are in trouble and you're going to find that you're not going to survive anymore. And actually cyanide poisoning deals with all of this, as cyanide poisoning blocks this electron transport chain and ultimately doesn't allow you to make any more ATP. So that was a lot of stuff, but what's the point of all of this cellular respiration? Just to make ATP. So start to go back, figure out how each of these steps work. Again, what goes in, what comes out, what happened with energy, right? Try to sum it in a way that you can understand. Good luck.